Okay, in the first video for this module, we will focus on the topic of parentage analysis, so how we can use molecular data to help us determine the parentage of different offspring. So this is important because we have these two general types of mating systems. They're explained in more detail in your textbook, but the idea is you have a social mating system, and that is inferred from direct observation. So say you have this nest, you see these two birds at the nest, you assume one is the mother, one is the father. So that's the social mating system based on observations. But there's this other type of mating system that's based not on observations, but on the genetics. So based on molecular data, we can determine who the actual parents of the offspring are. And so our genetic mating system reveals the actual parentage, which doesn't always match the social mating system, which is why it's important for us to use molecular techniques in this context. So this is a picture that I've shown previously when we talked about different types of selection. This male right here is considered a sneaker male cuttlefish, where what he's doing is on the side of his body facing the female, he's convincing that female, hey, I'm a male, I want to mate with you, but there's another male over here, and this sneaker male is showing a different pattern there and to say, no, I'm not doing anything, I'm just hanging out. And so this idea of having males, for instance, that are sneaker males, you can actually have them mating with females, which is what in some cases leads to this difference between genetic and social mating systems. So if you were considering this example of cuttlefish, you might have this one dominant male where you would assume that he has this territory. Any females in his territory, he's probably the father of her offspring. That would be the social mating system. But if you do genetics, you might actually figure out that the actual parentage for some of those offspring is this sneaker male. So using that molecular data can allow you to identify for sure who the actual parents are. And when we're doing this, often the parent we're not sure about that we're trying to figure out is the father. And so there's another example here on the right. We have a female, this is a midshipman fish. We have this giant garter male. You can see he's much bigger, but we also have a smaller sneaker male. And if you want a little trick to remember who we're often trying to figure out who that parent is that we're not sure about, you can reference Star Wars, the famous I am your father scene. My husband is a huge Star Wars fan. I'll have to ask him if I get some bonus points for including a Darth Vader I am your father reference in my lecture. So when we're doing these types of genetic parentage analysis, usually we know who the female is. In general, if we see a female at a nest site, for instance, we can be pretty sure she's the actual mother, but it's the father we're more unsure of, and that's what we're trying to determine. So one of the ways we can do this is using a process of exclusion to rule out potential putative parents and figure out who the actual parents are. So let's consider this example here with these robins. So say we have an offspring who has a genotype of big A, big A. That means that particular offspring had to receive that one copy of the big A allele from the mom, one copy of the big A allele from the dad. So we know that, right? If we also know that the mother's genotype, so we'll assume this mother at the nest here, this bird at the nest is the mother, and we'll assume it's a genetic mother. So the mother has an AB genotype, then that makes sense, right? We have no reason to believe that she is not the biological parent. We know that she can have passed on that A allele to her offspring. So let's say we're trying to figure out who the father is, right? So let's say this guy right here is the social father. If we know that his genotype is BC, then we know that doesn't make sense. That individual must have passed on a B or a C allele to its offspring. That doesn't match with what this offspring actually has. So we know that in this case, we can actually rule out this male. That social father is not the genetic father of this offspring. So in that case, we would have to look for another male, maybe in the surrounding area that we know does have at least one A allele in its genotype at this locus that it could have passed on to its particular offspring. That's what I mean by exclusion, is we can, based on a process of elimination with the genetic data we have, that's what I mentioned, I think this is kind of like a puzzle. We can exclude a particular parent based on the genotype of the offspring, the mother, and then the potential father. So here's another simplified example here. A lot of these studies, I'm using a lot of bird examples. A lot of these studies are done in birds because that's one of the main systems where they have established social mating systems, but also sometimes genetic mating systems that are different. So that's why I have so many bird examples. That's often the type of organisms that these studies are conducted on. So say we have an example here. Now we're looking at two different genetic loci. So say we're looking at an albatross little chick here that has these two genotypes. Locus 1, it has a big A, and at locus 2, it has AC. We also know the genotype of the mother here. She has an AB genotype at both loci, which we can infer then she passed that A allele on to her offspring. So what we know now is that these alleles are accounted for. They came from the mom. So what we're trying to figure out is where this chick got these alleles from, the A and the C. And so what we can do is look at three potential dad albatrosses, and we can figure out, based on information here, whether the social father is actually the genetic father or if it's a different neighboring male. So if we look at the genotypes for the social father here, the genotypes at locus 1 is AA, and at locus 2 is BB. If we consider the remaining genotypes that we're trying to fill in here for the chick, those don't match. So the father had to have passed on the A allele at locus 1 and the B allele at locus 2. The chick does not have a B allele at locus 2, so we can then rule out this social father. We know that this guy is not the actual dad, despite what the social mating system looks like. If we move on to neighboring male number 1, same kind of thing. He also only has two B alleles. That means he cannot possibly be the father of this chick because the chick doesn't have a B allele. So we can rule out that neighboring male as well. And then if we look at our neighboring male number 2, remember the genotypes we're trying to fill in for the chick that were not accounted for by the mother are the A and the C alleles. And if we look at that neighboring male number 2, he has both of those alleles present, so we can then conclude that neighboring male 2 is actually the father of this chick, not the social father. Again, this is a simplified example, so in the wild, you would have to use more loci, and you'd also have to sample more individuals. It probably isn't likely that you would find the actual father just by sampling 3 males, but this is overall how that process works. And when we're thinking about, again, we're using molecular data, and we can use high throughput sequencing, which we talked about a lot already this semester, to identify SNPs that can help us assign paternity and, again, determine whether there's a match between the genetic and the social mating systems. So this is an example from the variegated fairy wren, and I looked it up because I thought this bird was super cool. Unfortunately, they're only found in Australia, but this is the really striking color patterning for the male, and then these are the females. And in this study, what they did is they used different types of markers and compared them to figure out which is generally more effective or accurate when you're trying to determine parentage of different offspring. And the two different markers they compared are microsatellites, which are historically most commonly used in these type of parentage analyses because they are so variable. And they also use SNPs that were identified from ddRAD sequencing, which we've also talked about previously. That's a type of reduced representation genome sequencing. So they identified a bunch of SNPs, and what they did is compare the parentage results using the different types of markers. For the y-axis here, we don't have time to go into all the details, but the idea is that when you're doing these types of paternity assignments, what the programs will do is assign one male that is the most likely father. So it'll give you some type of assignment score for how likely it is that that male was the father. And then there will be some kind of break, and it'll give you other possible males that could be the father, but the probability of that is lower than that top choice male, right? And so what this graph is showing us is that it's the difference. So what this graph is showing us is that the difference here in between that top choice male and the other possible males that are the fathers is much greater for the SNPs. So what that means is that we're more confident in this father that we've chosen based on our data. We can be more confident that we're picking the right father when we're using SNPs versus when we're using microsatellites. So this is one example of how we can potentially compare two different markers, and overall it looks like SNPs are a better choice. And as I've talked about a little bit already in this particular video, oftentimes when we're doing these types of parentage analyses, what we're trying to do is determine whether extra pair fertilizations have occurred. That would mean there's a mismatch between our social mating system and our genetic mating system. So in this schematic, say we have a nest here with different eggs. WPO stands for within pair offspring. So that would be, say you have a male and a female bird that are taking care of the nest. They're the pair. So these two eggs are actually the offspring of those two parents that are taking care of the nest. This EPO individual or extra pair offspring was actually fathered by a different male. So maybe a sneaker male or something like that. And so parentage analysis can help us figure out whether there are these extra pair fertilizations within a litter or a nest. The terms are different depending on what type of species you're studying. And so they've done this for a lot of different species. There's a table in your textbook that summarizes some different examples. And here's just one example I thought was interesting that I pulled. They've done these types of analyses in fat-tailed dwarf lemurs, which is quite a name. And they found that in this particular study, 44% of the fat-tailed dwarf lemur offspring are actually a result of these extra pair fertilizations. So if you have a litter and you have a mom lemur and a dad lemur taking care of the offspring, 44% of those offspring are actually fathered by a different male. So that's one example of the types of information that molecular data can give us in terms of helping us figure out parentage, which is again under this umbrella of behavioral ecology.